Welcome. My name is Tristan Debona, and thank you for tuning in to SPEX SamplePrep presentation on the 2010 Geno Grinder and 1600 MiniG high-throughput tissue homogenizers. The 2010 Geno Grinder is an ideal solution for homogenizing most plant and animal soft tissues. This is accomplished using a bead beading method, whereby beads are placed in your choice of tube or tighter plate, and the vertical motion of the clamp at high speed creates the beading motion. Most samples can be sufficiently ground up or homogenized in 2 minutes or less using this method. Additionally, our non-proprietary clamp allows you to use a multitude of different sized tubes, vials, jars, and plates. Homogenization is the idea that blending or mixing dissimilar materials will create a uniform mixture where anywhere within that mixture or anywhere that you sample from, it will have roughly the same percentages as anywhere else in that mixture. The way to think of this is think about making chocolate milk. When you first start, you have the syrup or the powder separate from the milk. Once you stir and once you mix that, it eventually becomes uniform and tastes like chocolate milk from the first sip to the last. Here we use an example of yeast. Before and after, you can see the pelletized yeast separate from the buffer and the beads down at the bottom. After homogenization, you have not only the yeast being distributed and being ground up throughout the buffer, but you also now have sufficiently lysed cells where you can extract whatever you were looking for out of those. The idea behind homogenization here, of course, is to reduce the particle size, get a uniform particle size, and to distribute evenly. That way you can expose surface area and perform an efficient extraction. So what is bead beading? Well, bead beading is exactly what it sounds like. You're taking beads and using them to bead up or pulverize a sample and reduce the particle size. Different types of bead beading media can range from stainless steel to different types of ceramics, glass, silica beads, as well as garnet or other types of material, walnut shell, ceramic cylinders. They can take many different shapes, many different sizes. You can go anywhere from a ½ inch all the way down to something on the order of several microns. And how this is accomplished is you would place the beads in the tube with whatever sample or matrix that you wish to homogenize, and then by shaking vigorously, you're able to break up the tissue, break up the cells, release whatever molecules you're looking for. Some important things to note about bead beading is that the sample mass and the hardness of the composition are critical when determining what matrix or what grinding media and what tube, what size to use. This is also important in considering how much sample you may want to use as well. The more sample that you use, typically the larger tube that you will need and maybe the larger or heavier bead you will need. And what I've found is that mass is directly related to the grinding efficiency. So if you're grinding up very small tissues or cell cultures, things of that nature, then using small, fine beads is more than adequate. But if you're getting into larger, more solid or more fibrous material, you're going to need larger beads with higher mass. You may need fewer of them, but they will take up more room in the tube, so that is a consideration as well. Using this method, you can process your samples frozen. You can process them at room temperature. You can process them dry or wet, depending on the type of sample that you're working with and the special considerations thereof, whether you're looking for temperature stable molecules, which require lower temperatures, or if you're not concerned with that, any of these can be addressed using this type of method. You can pre-freeze, you can pre-cool, you can heat up, any of those sorts of things before going into the instrument and doing the grinding. Some other points on here is that the total volume of samples and buffers is typically limited, usually about 25% of the sample volume. And you can see there on the right-hand side of that picture where you have the tube, you have a liquid sample, a beads, and a grinding media in there. So that is more or less a general protocol when dealing with any type of sample in any type of tube. Is about 25% is the sample volume for an efficient grind. Too much sample will reduce the grinding efficiency. Likewise, not enough sample may create issues with the tubes themselves or with the grinding media, and you may get excessive heat buildup, you may get degradation or over-grinding, or you may get rupturing or damage to the tubes. Heat buildup is a concern. Generally speaking, in the Geno Grinder, what is really nice about this unit is that you're grinding things so quickly in such a short amount of time that heat buildup is really not that appreciable. This, of course, can be remedied in situations where it's critical to maintain a temperature by pulsing or grinding cryogenically, pre-freezing the sample. You can use liquid nitrogen, dry ice, -80 freezers, a regular freezer. There's really no condition there. Now I'd like to take some time and discuss the features of our 2010 Geno Grinder. One unique aspect of this unit is the adjustable clamp with the quick release button. This quick release button is on that orange top piece that you see there in the picture. It has a little button in the middle, and you press that in, and that allows you to slide it on or off that threaded rod without any additional work. This makes it very easy and very quick to remove the samples and change out formats or change to the next samples very quickly and easily. Now, once you put it on, you simply give that a twist. It's a kind of a triangular three-wing knob, and you give that a twist to tighten it down, and that tightens it and locks it in place. So a couple of nice things about this is that having this type of clamp design allows you to work with any format that you want. You can stack up to 6 titer plates of the 96 well or any other variety that is the same basic dimensions. You can use up to 16 50 mil centrifuge tubes using a foam holder you see in the bottom right. You can use 6 75 milliliter vials. You can use Eppendorf tubes, PCR tubes, micro Eppendorf tubes, 2 mil, 5 mil, and 15 mil tubes, and anything else that will fit in that clamp. All you need is a foam holder or some sort of material in there to prevent it from moving around in the clamp, and it'll run just fine. And as far as the clamp speed, so that whole mechanism, that whole clamp stack that you see there will vertically shake. It'll go in kind of a stroke motion between 500 and 1750 strokes per minute. Slower speeds are usually used to mix or blend samples, whereas higher speeds, usually anywhere from 1200 RPM up, are used for homogenization or grinding. Some additional features of our upgraded 2010 Geno Grinder include the fully programmable touchscreen, which now adjusts as well for the viewing angle. It has a data port that allows you to export your run history. It has a user lockout feature where you can password protect and keep your protocols from being changed without authorization. You now have the ability to save many protocols. It has a full screen of slots for different protocols to be saved in, and that includes all the parameters for the run. And now everything is digital touchscreen, so to input your run speed or amount of time, pauses, that kind of thing, you can do from the touchscreen. The new unit has the same rugged construction as the older units, but with improved soundproofing and an improved lid lock for safety. This translates to lower decibels in your lab, small footprint still that will fit on most, if any, countertop, It still has 115 volts, so it'll run at a regular outlet with no special power considerations. It has rubberized isolation feet as well as a full suspension system inside for the motor and the assembly that minimizes the amount of vibration translated through to the countertop and the rest of your surroundings. And in addition to that, our new system has a LED light for improved sample visibility while it's running and a large viewing window to better accommodate that. And now we have a little bit more detail on the touchscreen control panel. How this differs from the previous knob sets on the older models is now you have everything on a touch screen, and you have some additional features like the rest and the cycles. This allows you to pause, run short periods or run long periods and pause and pick back up. It also allows you now to store up to 500 protocols. So different types of material, all the different samples you may be working with, you can name them and you can store them for easy one-touch access. As we can see in the top left, up to 5 cycles with rest times can be set. This will allow you to minimize temperature increase while grinding, especially on extended grinding protocols. There is the administrative lockout feature that I've mentioned before, which allows you to restrict user access and protect those protocols from modification without authorization. Run history is recorded for every single run. This can be manually cleared, of course, but it can also be exported via USB or USB cord to your desired format. And other resources on the control panel now include instructions. The full manual is uploaded on there, as well as a catalog of all the accessories available for this unit. Some of the unique accessories that we offer for this unit include our full line of Biotech accessories, which are essentially heat sinks or cooling blocks that are made out of metal, and they either click into your titer plates, as you can see down there kind of in the bottom center, looks like a big aluminum heat sink, or in the back of that you see the ones with the tubes in them. Those are hollow blocks for the most part that allow you to chill the sample in liquid nitrogen, dry ice, or freezer, and that metal will act as a heat sink during the operation and draw heat away from the samples as they grind, effectively stabilizing the temperature of your sample for the short period in which they're in the instrument. You'll also see on the side, on the right-hand side there, our 2600 cryo station, which is a liquid nitrogen cryogenic treatment station that allows you to chill those blocks and the samples in a nice benchtop format right next to the instrument for quick and thorough cooling, as well as immediate temperature stabilization both pre and post run. In addition to those, the Biotech accessories, we also carry a wide range of different grinding vials for specialized applications, including preloaded 2mm tubes. We have some cryogenic tubes that you see in that picture there that include tungsten carbide beads. And we have some polycarbonate and some kind of specialized grinding vials and tubes as well. And in addition to that, we do sell all different types of grinding media, everything from steel balls, molecular grade or molecular biology grade, acid washed beads, to ceramic cylinders, as well as full line of different types of ceramic and silica media as well. As briefly mentioned before, we do sell a full line of foam holders for all those different formats that we discussed. We sell titer plates and cap mats in different formats, everything from 96 well to 24 well, round and square wells in different formats. And we have a convenient ball dispenser. The 2100 allows you to dispense one 4mm bead per 96 well. And if you need more than that, you just hit it a second time. Very fast, very convenient for those working high throughput with titer plates. That's a great device. So who can benefit from an instrument like this? Well, currently these are in use in the biotechnology fields, everything from bioengineering, medical kind of research, pharmaceutical research, agriculture, crop science. government safety, testing labs, contract laboratories doing all sorts of different things, food safety, environmental, even some clinical labs doing blood, hair, toxicology, universities, cannabis testing, which is a big market right now for this instrument, and there are many more applications that I'm not mentioning. Basically, anyone who needs to grind anything of a softer nature can benefit from this instrument. So the Geno Grinder was initially developed with crop science and agriculture applications in mind. Applications that needed high throughput to process plant clippings, anything from stem, leaf, seeds, rice, animal feed, any of those kind of agricultural products, and prepare them for either DNA, RNA, protein extraction, in some cases regulatory things like mycotoxin or pesticide, heavy metal testing, those kind of things. And it was developed with their needs in mind because they really needed something high throughput. In this industry, you have a lot of large batch sizes, large sample volumes, and continuous workflow. So this may go on. You may need something that can process 96 well plates, 6 at a time, 24 hours a day. And thousands upon thousands upon thousands of samples. And that's where something like this can really be beneficial. For one, it provides consistency, and it really streamlines the laboratory workflow by doing multiple samples and by doing them quickly and by doing them consistently and efficiently each time. Getting to things like RNA and proteins, these are things that are temperature sensitive. They can break down very easily as soon as a temperature reaches a certain threshold. So we have the cryo blocks to protect those, even when you're doing hard things like seeds or fibrous material, which can be difficult to grind and can take longer than the 2 minutes that we talked about earlier. Those cryo blocks will protect it well up into the 5-minute range, which is more than enough to process that material. This can then be used in various steps using the protein isolation, RNA, and DNA isolation kits to isolate those genes or gene products that can then be analyzed by further methods. So, getting into some application specifics, we have leaf tissue. Leaf tissue can range. You can have very different types of leaf tissue, some that's more fibrous than others, dry versus wet leaf tissue, or fresh versus dry if you're talking about tobacco or cannabis, and others. So leaf punches are typically used for genetic analysis. This is a very common procedure in the field. They're easily homogenized in this unit and other bead beating units. But this one particularly very quick and very high throughput. So if you have a lot of leaf punches to do, small samples of 50 milligrams or less can be processed in the deep well titer plates, which are the ones that you saw in the previous pictures, or in 2 mil tubes. And if you have larger sample sizes, you can go up in size to 5 or 7 mil tubes. Using the stainless steel or zirconium oxide grinding balls. The denser the leaf tissue and the more fibrous the leaf tissue, the more beads you're going to need. So typically, if you're doing a softer material, then 1 or 2 of those 4 millimeter stainless beads is all you're going to need. If you're doing denser or more fibrous tissues, then add more beads. Typically in the 2 mil tubes and even in the titer plates, I believe you can get up to 3 beads per well in the titer plate safely, and about the same 3 to 4 in the 2 mil tubes in most of those standard conical bottom 2 mil centrifuge tubes. 100 to 200 milligram samples are going to require a larger vial. Typically they have 4 mil on the slide, but typically you could see anything from 4 to 7 depending on who your distributor manufacturer of those is, with 4 millimeter and up to 9.5 millimeter grinding beads. So more difficult tissues, again, fibrous tissues, things of that nature, you can use a 9.5 millimeter stainless steel grinding bead. You'd use only 1 of those per vial. And if you're doing up to 1 gram of leaf tissue, you can do it in 15 mil polycarbonate vials. You can do it in 15 mil standard, I believe it's PET or PEF centrifuge tubes. and you would use, again, either the 9.5 millimeter grinding beads or a combination of 9.5 and 11 millimeter stainless steel grinding beads. And that's because you don't, if you have too large of a bead in the tight cone at the bottom, you may compact the material. Another thing to mention here is that when you're loading these, you would pre-weigh the material. You would add probably a bead to the bottom of the tube before adding your sample, then add your sample, then put another bead on top. That'll help prevent that compacting, and that'll get everything between those beads to homogenize quickly. So here we have a crop science sample protocol, or what we would call workflow, and this would be how to homogenize wheat seeds. And you can extrapolate from this for other similar seeds. You'd place 1 to 2 of the seeds in a 2 mil preloaded tube, and here we give you the part number for a couple of different options. I believe those are the options that are preloaded with stainless beads. You can also use tighter plates, and basically the preload for the 2 mil tube would be similar in that tighter plate. Add 250 microliters of your lysis buffer. Again, that applies to both the 2 mil tubes and to the tighter plate. Place the capped tube or the capped plate into the foam holder or directly into the clamp. Insert it into the clamp and tighten the clamp down on top, which would be pressing in that quick release button, sliding it all the way down till it makes contact with the tops, and then giving it a little snug with that orange knob. Set the speed to 1500 strokes per minute, and I'm going to do a little side note here that 1500 is kind of the sweet spot for most material. Under certain circumstances, you may have to tweak that. You may want to go a little slower. You may not need the full 1500, or you may need to go a little higher. But typically, I say 1500 is really a good kind of maximum RPM to look at. If you're looking for something to start with, start at 1300 and work your way up. But 1500 is more or less, that's acceptable too. If you want to just start there and go from it, you'll probably have success. Set the runtime here for 2 minutes, and then once that is ground up, it's ready for the next step in your extraction method, whether that be a cleanup, isolation, precipitation, whatever it may be. And some of those steps can be performed in the Geno Grinder as well. You'll find that once you've done the homogenization, a lot of the other steps may require shaking or mixing. You can do that at a nice low RPM using the Geno Grinder. So you can simply uncap it, add or do whatever you need to the sample, put it back in, so on and so forth. So it's nice and versatile in that regard. Another method that the 2010 Geno Grinder is uniquely suited to is the catcher's method. Some of you may be familiar with this. It's been around for quite a while and has become quite a popular method for the analysis of pesticide residues and other organic contaminants in food and beverage products. It's a very easy, simple, non-time consuming, non-labor intensive method meant to accurately extract these kind of molecules, but also using pre-measured packets and pre-made kits so that it makes the lab tech's job very easy, very simple, and they can be very high throughput. It stands for quick, easy, cheap, effective, rugged, and safe. Samples are shaken in a centrifuge tube with a mixture of salts and solvent, and then you can do that in the Geno Grinder. Following centrifugation, you do a cleanup step that again uses the Geno Grinder to shake the sample, and then the analysis is simply completed using GCMS. We've done several tests and several studies using the Geno Grinder to measure the difference between mechanical shaking and manual or hand shaking. You can see that 1 minute in the Geno Grinder is more than sufficient to break up the material to a good degree. The hand shaking method alone is very inefficient. You can do a limited number of samples at a time, and it's going to vary wildly depending on who's shaking, how long they're shaking for, how vigorously they're shaking, the stroke, how long their arm reach is. Whereas in a mechanical system, you can do 16 50 mil tubes, 24 15 mil tubes, or 96 2 mil tubes at a time. And you're talking about a 1 minute, 1 to 2 minutes. This improves the consistency, making sure that the agitation is consistent from one sample to the next, anywhere within that clamp, and one run to the next, meaning it's the same every time you run that instrument. Make sure it essentially ensures that the conditions are reproducible each time. This also yields an increase in recovery. The more vigorous and more accurate shaking of the mechanical grinder is going to produce a more thorough extraction. And there are many different methods and protocols that also reference this kind of mechanical grinding specifically for their method, such as the catcher's method we spoke about earlier. So it's been validated multiple times over, and it is in use and is even recommended by certain manufacturers. And you can find some of these catcher's kits available from suppliers like UCT, Agilent, etc. Now we can discuss a little bit about the broader range of biotechnology applications. As we mentioned before, animal and plant tissues are what this is optimally designed to homogenize. So cell lysis, extraction, mixing of samples with reagents at slower speeds. But this is also great for working with yeast, microbial, bacterial, fungal cells, things of that nature. Seeds, whether it be hard seeds like soy or corn, any type of tissue in there is really, there's a way to break it up in this instrument. It really works well for most tissues, excluding things like bone and cartilage, but pretty much anything else. This can also be used on direct biocatalysis kind of things. So if you are breeding E. coli and looking for an expression product in there, or doing it in mammalian tissue or anything of that nature, breaking up those cell lines and the cell lysis to get those products out without damaging the products, that can be accomplished in this unit as well. Total RNA, DNA, and proteins, these can be isolated from the animal tissues or from the cell lines, obtaining those genes and gene products. If you use the instrument correctly, if you do a little experimentation, you'll find that you can find that kind of that nice area where you get great DNA extraction without too much shearing or degradation. RNA is great. It's already a small molecule, so it's very easy to isolate, and provide that you keep the temperature stable with the cryo blocks, you have no problem isolating that and pulling that out. And we have lots of customers and lots of applications now, people using this instrument to pull out RNA, microRNAs, tRNAs, all sorts of different RNA products. And here we have a basic biotechnology workflow or protocol. So to homogenize the tissue, you'd place a certain amount, 50 to 100 milligrams in this particular diagram, of the animal or plant tissue in a 2 ml preloaded vial. Add the 500 microliters of lysis buffer. Place the cap tubes into the foam holder, and then place that foam holder into the clamp as we did before. Set the speed to, this says 1,000 strokes per minute. You can go up to 1,200, 1,250 strokes per minute, depending on the nature of the tissue, whether it's softer or it's more fibrous. And you can do that for about 2 minutes, and then the samples are ready to move on to the next step in extraction. Now you'll find here that even though we give kind of specifics here, this is using animal tissue. You'll find that in the biotechnology field, you're going to have a lot more variation in the grinding media. So you're going to have everything from those silica beads for cell lysis all the way up to those stainless steel beads for tissue lysis. And once again, when picking your accessories, we like to reiterate that it's very important to match the vial type and the grinding media to the sample type. So for hard samples, things like seed and things of that nature, we're going to recommend using 2 mil or 15 mil short tubes with larger steel balls. Basically, you want those balls to have less area, less height to travel, and you want them to have more contact time with the material. For soft samples, things like algae, bacteria, like yeast that we mentioned before, cell lysis applications, we're going to recommend using 2 mil or 15 mil tall tubes with smaller silica grinding beads. Silica or some maybe of the zirconia ceramics. Cryo grinding is typically recommended for these as well, especially with RNA extraction. The most important thing is not to overload the vials. Too many grinding beads, too much media, and you will not get an efficient grind. Also, you can risk rupturing the tubes and having a leak and contamination within the instrument. We do have specific protocols available on request for the Geno Grinder from DNA kit suppliers like Zymo, Omega, Biotech, and a few others. And now, for the last part of our presentation, we'll discuss the 1600 MiniG. The 1600 MiniG is essentially the same as the Geno Grinder, except it is a scaled down version. This is a great option for people who don't have quite the high throughput needs of the Geno Grinder, but still need something with its capability and the capacity to do all of those different formats. So it's got a smaller footprint, but essentially it goes everywhere. It does everything that the Geno Grinder can do, just with lower throughput. The 1600 MiniG is optimally suited for preparing plant and animal tissue samples for testing, as well as cell lysis. It uses the same vertical pulverizing motion to ensure complete and uniform sample processing, has a maximum speed of 1500 strokes per minute. So it does not go quite to the same speed as the Geno Grinder, but as I stated before, most things will grind up at that 1500 strokes at a maximum. So that it will do pretty much anything that you can do in the Geno Grinder. The clamp will support 2 titer plates stacked. It'll support 6 50 mil centrifuge tubes, 6 15 mil vials, or 48 2 mil vials. We do also offer a full line of similar accessories scaled for the MiniG. So we have the same range of foam holders, grinding media, as well as cryo accessories that will adapt to this unit as well. That concludes our presentation on the 2010 Geno Grinder and 1600 MiniG tissue homogenizers. On behalf of SPEX SamplePrep, I'd like to thank you for tuning in. I hope that you enjoyed this presentation. Should you have any questions or require any further information, there will be some links and contact information in the description below on this video. Thank you very much. Have a wonderful day.